It is such an honor to be here. I especially want to thank Perth NRM, Regen WA, and Greenmate for bringing me out, and especially all of you for welcoming me to this beautiful city. The International Panel on Climate Change, their last report specifically points out that soils is kind of one of the easiest and most manageable ways for us to sequester carbon. Soils harbor a big portion of the planet's biodiversity. Communicable and non-communicable diseases, how it is directly related to the soils and the soil microbiome. It's kind of no wonder that soils has become so important. Sustainability reporting right now is a checklist. So when we talk about human health, we have certain general recommendations for everybody. And that's kind of what our sustainability has right now. Joel did a great job of going through those things that everybody can be doing to improve the microbiome. And yet, we're still recommended to go to the doctor, because there's an individual component of that as well. And he's actually looking at specific measurements. He's looking at metrics associated with me. And so he's giving me some feedback of things that I need to be doing specific for me, for my health. So I've kind of listed through there a lot of the different actions that you can take in order to be able to mark those checklists of that sustainability platform. What is it that we need to do to be able to really measure soil biodiversity? So this was specifically looking at soil biodiversity globally. And so we came up with this big list of all of the things that we can be looking at. A lot of those have been discussed in this throughout this conference. How do we go from those actions that we're implementing? How do we go from talking about no-till agriculture? And how do we transition to that to say, are we actually gaining those benefits that we think we're gaining? You know, if you get it right, it's great. And if you get it wrong, it's a bumpy road. How do you know if you're getting it wrong? So we want to look at that and we want to say, based on those outcomes, based on actual measurements, how can we tell if you're doing it right or if you're going to get it wrong? The only way that you can really get to that is by saying, well, in this system over here where they've done the research, in the academic farms, in the plots, and hopefully that will apply to my fields, right? What we want to do is we want to bridge that gap, and we want to say everybody should be able to do that same kind of test, that same kind of information. We do this metagenomic or amplicon sequencing. And basically what that is we extract all the DNA from the soil and we sequence the DNA using amplicons. I believe that Craig did a really great job of explaining this. We're just taking a short little piece of the DNA. That little piece of the DNA has enough information in it to tell us who it is. These are all of the different individuals that you have, and this is the abundance of each one of those individuals. The number of rhizobium that you have, and this is the number of different types of rhizobium you have. We have that big, long list of what is all of the organisms that you have in your soil. And then on the other side of this component, we've done a lot of genome sequences on rhizobium, and so we know what they can do already. We know what bacillus can do. We know what actinomycetes can do, right? So we have all of the function that's available with all of this, and then we just put those two together. When we can do several different functions, we can look at not only phytohormones, but nutrient cycling. We can look at pathogens. We can look at not only the organisms that are, for example, making phosphorus more available, but also the organisms that are competing with the plant for that phosphorus. What are the pathways that are making it more available? And what are the pathways that are pulling from that available pool to make it unavailable? And so it's not just saying, I want to look at what's available to the plant, but what are the organisms that are there making nutrients more available to the plant? We're able to look at it from the perspective of disease cycling, of disease suppression. We do have that evidence over here, and we're relying on that evidence over here, but we want you to be able to have that evidence for your soils, for your field, so that you can look at it and you can say, this is what my soils are doing. And so then we're able to kind of say, this is where your soils fall. And this is kind of an acceptable range. This is where we see good growth happening. And is that a good value or is that bad? Is it high? Is it low? And so we're able to do that same thing and look at all of these different diagnostics from the biological perspective of the soil. It's a way to be able to really measure what's going on. And the benefit of this is that you're able to get that feedback. You're able to say, is this working? Is this moving the things that really need to be moved?